this is a good example of the lacunae. You can see the space, a little white area. You can see the cell sitting right inside of it. Okay, perichondrium is your outer layer of cartilage, and the hyaline cartilage is inside. It's composed of lacunae with chondrocytes, and then the matrix around it. All right. See if my computer can catch back up again. Same thing again. The perichondrium is your epithelial layer. The chondrocytes, which produce cartilage. And the matrix around it. And the chondrocytes are in lacunae. Okay. Magnified view. Again, you can definitely see. Oh, where was back here? There he is. You can definitely see the lacunae in there. Okay. Good boy. All right. Okay, this is your pubic symphysis, fibrocartilage. You want this to be flexible, especially in females. This is where the birth canal allows the bones to stretch as the baby comes through the birth canal. So this is your fibrocartilage here. Okay, you can see bone tissue on the bottom left. You can see the cartilage inside the center here, the fibrocartilage. Again, we haven't talked about that yet, but we will. We will get into that. Okay. Again, see the matrix. Matrix is what's between the chondrocytes. Then you see the chondrocytes inside the lacunae. Okay, this is between your vertebrae. We haven't talked about the vertebrae yet, but on your vertebrae, the bones don't hit bone to bone. There's what's called a cartilaginous pad in between each vertebrae in your body to cushion when you walk, when you jump, when you fall, whatever. It cushions the blow between the vertebrae so they don't hit each other. Okay? When that blows out, the bones hit bone, that's when you have a problem. Okay? And you have a ruptured disc, things like that. This is what the cartilage is. This is a cartilage in between the vertebrae. Okay? It's the same thing. It's got a matrix. It's got the chondrocytes inside lacunae. But this is your intervertebral discs made of the cartilaginous pads that separate your vertebrae. And again, we'll get into real detail on those when we go into those other chapters. Okay, this is the epiglottis. It has got elastic cartilage in it. Okay? That allows it to withstand forces in it as it expands when food comes through. Okay. Perichondrium is the outer layer. You have your elastic cartilage, and then the chondrocytes in your lacunae. Again, same thing we've been looking at. The matrix is between them. Chondrocytes are in the lacunae, and this is elastic cartilage. Allows it to expand. And the perichondrium is your outer epithelial layer for it. Yeah, you see the elastic cartilage, perichondrium, lacunae with the chondrocytes. And we'll go into this a lot more detail. Okay, look at the difference now. This is bone. This is not cartilage anymore. Cartilage has all those fibers that run through it. Bone is these individual hard-packed cells. These are called osteomes. Osteome is the bone segment of where it's produced. Okay? You have a central canal. You have perforating canals. Bone is perforated by arteries, by veins and by nerves. Okay? There's nerve cells inside the bone that go through it. That's not the same with cartilage. Cartilage is avascular. When you get your ears done, your ears pierced through cartilage, okay? It doesn't hurt. I mean, comparatively. When you break a bone, have you ever seen a patient with a broken bone? It hurts when you break bone. There's nerves inside the bone. They run through these perforating canals, and they're accompanied by blood vessels. Right? Arteries, veins, and nerves all run together. Okay? Anybody give injections to patients? You aspirate certain times, right? Yeah, there you go. Why do you aspirate? To make sure you're not in a blood vessel. Okay, because you say you want to give a nerve block in dentistry. You want to block the inferior alveolar canal, inferior alveolar nerve. You put the medication right there and you aspirate, because if you don't want, you have blood vessels running right by the nerves. They all run together. You don't want to put local anesthetic into the nerve. Local anesthetic has epinephrine in it for dentistry. Epinephrine does what? Epinephrine is adrenaline, right? Okay, it increases blood pressure, makes you sweat, makes your heart beat fast. Not good things in a dental chair. People are nervous enough. You don't want to make them more nervous. Okay? So you gotta be careful where you inject it. So you aspirate, you pull back on the syringe, you make sure blood does not come through the needle back into the carpel. If it doesn't, then you can inject around the nerve. But they all run together. The bone is very vascular. If you break a bone, it bleeds. If you break cartilage, it doesn't bleed. Okay? So this is what bone looks like. Again, you have your lamellae. These are lamellation, called lamellae patterns. They're circular patterns. Okay, remember when we looked at the cartilage, we saw these wavy lines that went through it. Bone is circular patterns. It's called lamellae, lamellated patterns, which means circular patterns. And this is compact bone. This is your outer bone. This is very dense bone. Okay. That's going to lag again for a little bit. Let it catch up. Okay, so you have these circular patterns. Your osteome is one circular pattern. This is an osteome. This is an osteome. This is an osteome. It's a circular pattern of bone. These are the osteocytes. Osteoblasts. Osteoblasts produce bone, just like chondroblasts or fibroblasts produce fiber tissue. Okay, this is your central canal, and these are the lamellae, the concentric circles around it. Kind of like the rings of a tree. When you cut through a tree, you see the rings. That's kind of like the bone here. Very similar. Okay, so here's your central, your rings around the bone. This is your osteome. Canaliculi. Canaliculi are very small canals. So it's canaliculi. They're very small canals, and they carry your blood vessels and nutrients throughout the bone section here. These are your lacunae. Inside the lacunae are your osteoblasts that make bone. Okay? That's the lamellae, the concentric patterns, and your central canal. Okay? This is in compact bone. Okay? This is developing bone. You don't have the patterns yet. You have bone marrow. The periosteum is the outside layer. You have what's called trabecular patterns, or trabecula of woven bone. Trabeculation means like a spider web. When you look at an X-ray, you'll see crisscrosses within the bone. That's a trabeculation pattern. Okay, that's inside the spongy bone or the inner part. The cortical bone or the cortex doesn't have trabeculation. Okay, the medulla does. And trabeculation is just like a spider web. It's all these little weaves of bone. And you can see it down in here as it goes through. I don't think we have any X-rays here to show it. But again, you see bone. What you have, remember the other side, you saw all this bone, it was solid. It was all these concentric circles. You look at this, okay? This is not solid anymore. This is bone here, bone here, bone here, and we got spaces in between there. So what forms a spiderweb image on an X-ray? Okay, the bone marrow is the inside. Periosteum is the outside. And this is the spongy bone. This is the inner portion. It's not going to be as dense as your cortical bone. Okay. Got to catch up again. Okay, endosteum is the inner portion. Okay, the periosteum is the outer portion. Endosteum here is your inner portion around the bone. These are your osteogenic cells. Okay, this is the bone marrow, the inner part, your trabeculation. See how, look at the difference between this bone. See how it's formed all this space here? This is bone. Okay. And the other bone with all these small circles, very, very compact. This is more spread out. Okay? So this is your trabeculation. This is your spongy bone. The other one was your compact bone. Spongy bone's a lot weaker, because that's where all your bone marrow is. It makes bones still have their strength, but gives it lighter weight. So your bones don't weigh 600, 700 pounds. Because the inside of your bones are more hollow. They're trabecular patterns, a lot of space. Okay, that way you can still function. If they weren't, they're all solid, you could lift your bones off the ground. Okay, it gives the bones their lightweight capability and still provides strength. And again, blood vessels run throughout bones all the place. Osteoclasts are different types of cells. Those are cells that break bone down. They produce calcium in the body. Okay, they don't live in lacunae. Lacunae are osteoblasts. Those are what produce your bone. Osteocytes destroy bone. They produce acid. So your bone is your storage for calcium. Okay. Remember, we haven't talked about muscle contractions yet, but we will. The two things required for muscle contractions are calcium and ATP. Remember, your heart's a muscle. For your heart to contract, it has to have calcium and ATP. If your body doesn't have calcium, where is it going to take it from to get it into the muscle so muscles can contract? It's to take it from your bones. Okay, that's why you have osteoporosis. That's the other kinds of things breaking down. These osteoclasts break down the bones, so calcium becomes available for muscle contraction. Because you have to have muscle contraction or you will cease to exist. Your heart has to contract. So if you're low in calcium, it's going to take it out of your bones. That's what causes all kinds of degenerative diseases with your bone structure, like osteoporosis and things. Okay, again, look at the difference. Okay, I want to get rid of that. Can't get rid of the. Can I hide the labels? There we go, just for a second. All right. So look at the difference. Remember, compact bone had the bone in these circles stuck together next to each other, all right on there, no spaces, very thick. Look at this. This has spaces everywhere. Okay, you look at all the space around it. This is trabecular bone or spongy bone. It forms a web-like appearance if you look at it on an X-ray. Okay, what are all these little white cells around it?
Fat cells.
Fat cells, adipose tissue. Perfect. How can you tell? There's no nucleus. Okay, all the nucleus is squished to the side, and it's just white cells. Some can be yellow, can be white or yellow, with this fatty bone where if it's yellow. All right, so you have this again. Everybody can, what I want you to be able to tell the difference is between trabeculation bone or spongy bone and compact bone. There's a big difference. If you look at this, you can easily see those are spiral patterns, bones spread out, there's spaces in between them. It's spongy bone. If you look at the other one, it's compact, had all those concentric lamellations around it, all compact circles. Okay. Okay, these are your osteoblasts right here. Okay, and that's the one thing that is similar. Well, there's a couple of things that are similar we'll go over. But one of the big similarities between cartilage and bone is lacunae. Lacunae is just spaces, little depressions in the bone where the cells live. Osteoblasts for bone, chondroblasts for cartilage. And again, you can easily see this is not a circular pattern. This is a trabeculation or spongy bone that's spread apart. There's no concentric circles like there is in compact bone. That's how you can tell them apart. Spongy bone. You have the epidermis of the skin, and you have different sections of the bone. Diaphysis, okay, is the center of the bone. Bones grow. Bones grow by apposition at the diaphysis and the metaphysis. The epiphysis is the end. We haven't gone into any of that yet, but we will. Okay, so the diaphysis is your bone shaft. And you can see the patterns of bone. You can see bone here. You can see bone here. You can see the trabeculation pattern here. And this is cartilage. Bone grows at what's called growth plates. Okay, at these plates where the epiphysis meets the epiphysis meets the diaphysis, you have bone growth. So bones grow on the ends and expand. They don't grow from the center. Okay, that's why kids that break bones can sometimes become stunted with their growth because they damage the growth plate. If the growth plate is damaged, the bone won't continue to grow lengthwise. So this is your growth plate right here, where these portions meet. Like I said, we'll go over the epiphysis, the diaphysis, and the metaphysis. And the skin covers it here. Like I said, if you break a bone, it is going to be painful because blood vessels and nerves run through that bone. Okay, and these are the different sections of calcification, ossification. This is an ossification zone. Ossification just means bone growth. This is your bone growth plate. This is cartilage, and the cartilage is being replaced by bone. Okay, this is your zones here. We'll go through all these as we go through it. But this is cartilage, and this is your bone being formed. See, it's like a hybrid zone right here, which starts to calcify. So everything's laid out in the embryo as cartilage. That's why babies are so flexible. I mean, you got to remember, they're going through that birth canal. Their arms and legs. I mean, have you ever taken an infant and taken his arms and go, woo, or his legs? I mean, they bend like Gumby. I mean, they do. They're cartilage. There's no bone ossified yet. Now, as they develop, that cartilage turns to bone, and those joints become a lot less flexible. Okay, but that's what this looks like. This is all cartilage. This is bone, and this is the ossification zone and calcification zone where it starts to turn into bone. And it occurs at these plates right here, at the epiphyseal and diaphysis, where the metaphyseal plate meets it right there. And like I said, don't worry about that all yet, because we will go over all that in great detail. Blood cells. Okay. There are different types of white blood cells. There's neutrophils, monocytes, lymphocytes, eosinophils, and those are all involved with your immune system. Okay, and involved with how body responds to inflammation. Each one has different functions. Most of your blood is red blood cells. Red blood cells you can tell because they're red, for one thing. But what can you tell me here? Look at these red blood cells. What's the difference between these red blood cells and these white blood cells?
They're filled in all the way?
Perfect. They have a nucleus. They're filled in. Look at the white blood cells. All the white blood cells have nucleus. Some multilobulated. Some have 3, some have 5. And again, we'll go over all that. But white blood cells have a nucleus. At least 1. Some have 3. Red blood cells have no nucleus. Remember, the function of a white blood cell is to reproduce, to fight infection, to fight viruses, to fight pathogens. That's what a white blood cell does. It's our immune system. They got to have nucleus to reproduce. Red blood cells have one function in life: to carry oxygen and CO2. That's it. They carry oxygen. If we had a nucleus there, it would take up room. You don't need it to take up room. We need more hemoglobin. So there's nothing inside a red blood cell except hemoglobin. They don't last. They last 120 days, and then they die. Then they get replaced. Okay? So red blood cells, white blood cells can last for years. You get memory. We get vaccines. Vaccines have our white blood cell systems have a memory of the virus it's exposed to. So now we can replicate those antibodies and produce a vaccine that we won't get sick from a certain virus. Okay, red blood cells can't do that. Okay, white blood cells can live for a lot longer time, but red blood cells 120 days and they're done. There's no nucleus, there's no mitochondria. And mitochondria's main function is to use oxygen to produce ATP, right? That's what mitochondria do. That's what we talked about. Why would you have mitochondria in something that's going to deliver oxygen? You don't want something that's going to use it up. Red blood cells are strictly hemoglobin carriers. That's it. No nucleus, no mitochondria. They live 120 days and they're gone. They're designed strictly to transport oxygen and carbon dioxide. Whereas the white blood cells have a nucleus and our whole immune response. But that's how you can easily tell the difference between a white blood cell on a blood smear and a red blood cell. Okay. More blood smears, and we'll take a look at them. Again, look at the red. Everybody can see the red blood cells, right? Things going to lag a little bit. Let it catch up. All right. White blood cells are very easy. They got nuclei in them. Red blood cells, no nuclei. Platelets. Platelets are little fragments that break off what's called megakaryocytes. They're these really big cells that just break off these little sections and send to your body. What happens if the body is traumatized, if there's a cut, if there's an opening in the bloodstream, these platelets adhere to it, and they help with the initial plug to seal the clot. Now, if your body makes too many platelets, it's going to start sealing your arteries, arterioles, which is not good. That's what causes strokes. That's what causes a lot of embolisms, things like that. The platelets stick together. Okay? The plates are your main defense, your first line of defense against an opening or cut in your bloodstream. They're the ones that clog it or seal it up. Okay, eosinophils are a type of white blood cells, and platelets you can see floating throughout. That looks like it has like swimming goggles on it, huh? If I can see it again here. Yeah. Looks like the goggles on this one here. You can see the nuclei. But that's a white blood cell. These are red blood cells, and these little dots in here, all these little dots are platelets that float around in the bloodstream. Most of your blood is red blood cells. Okay. Some white blood cells. We'll get the percentage and everything when we actually do that. Red blood cells. Okay, so everybody right now should be able to identify red blood cells, white blood cells. I don't care what type yet, but we will later on. Red blood cells, white blood cells, and platelets. Vastly different. Okay.
So why are some of the red blood cells white?
You got to remember the shape of a red blood cell is a biconcave disc. So it comes out and swoops in. Depending on how you did your section and how the red blood cell gets cut through the section, some of it shows the inner portion, which is very thin. Some shows the outer portion, which is very thick. So it depends where the red cells are being squished and stained. Okay, it just depends how they're rotated. Okay, so if you see like this one here, you look at this one here is rotated, so the inner portion, biconcated, is right at your face. You're looking right at it. So the edges are dark. If you're looking at this one here, it's rotated, so the darker end is rotated towards you. Okay? Got to remember, you got a three-dimensional object, and you're putting it on a two-dimensional plane. Does that make sense?
Yeah.
Okay, perfect. Yeah, it's very hard to do a lot of times. Really, it's a two-dimensional object. You're looking at two dimensions of it. Okay, again, red blood cells and white blood cell, which is going to have to be a monocyte. Monocyte is a phagocytic white blood cell that eats other cells. But again, you can see the red blood. Just as you pointed out, it's real important to notice some red blood cells are dark red, some have white areas in the center of them. It all depends when you squish that cell. You got to remember, it's elongated disc. In the center of the disc, it comes in real narrow. Okay, so if you're looking at it this way, you're going to see white. You're looking at it this way, you see all red. You got to remember, you're lining it with a three-dimensional object, and you're squishing it and putting it in a two-dimensional field. That's a really good question. And one more blood smear. In neutrophils happen to have three nuclei. You can see the three nucleuses there. Three nuclei there. You can see the platelets. These are platelets. And these are red blood cells. Okay. Perfect. Let's go back. We did connective tissue. Let's do muscle tissue. We did muscles and nerve. We talked about a little bit too. Muscle tissue. We only have 16 slides of these. All right. Look at this muscle tissue. Can I make it bigger? Yeah, I can make it bigger. All right. What is this muscle tissue? It says it up there too. So skeletal muscle tissue. How can you tell it's skeletal?
The striations.
There you go. Now I blew it up. Now you can see it, right? Everybody sees the striations. Look like fish bones. Okay? The perimycin means outside, peri means outside, endomycin means inside. But everybody sees those striations, right? So if you have all striations and nothing breaking it up, it's skeletal muscle. Is skeletal muscle voluntary or involuntary?
Voluntary.
Voluntary. Those are all your voluntary motions. Your arms, your legs, your locomotion, swallowing, everything like that is skeletal muscles. Okay? Okay, again, what kind of muscle is this?
Skeletal.
Skeletal muscle. Okay. You can see the striations going through here. Those are your bands. What happens is those striations become closer together when the muscle contracts. They pull those striations together. We'll talk about that with actin and myosin and the different Z bands and H bands and I bands as they pull together to get into the muscle part. But all the nuclei are lined up outside the bands. They're all lined up multi-nuclei for your cell. So each cell has multiple nuclei. Okay. Again, cross section of it makes it a little hard to identify, but you can see the skeletal muscle fibers here. If you do a cross section look at it, it's not easy to see the striations, but on your exams, I will definitely show you striations. You will see striations once you identify skeletal muscle. So striations, skeletal muscle fiber, multiple nuclei all lined up. Identify a skeletal muscle. Striations are easy to see. Okay. If you see a certain structure within the striations called an intercalated disc, then you got cardiac tissue. And we'll go into that. That'll show you that too. But again, you can easily see the striations, easily see multiple nuclei for this cell. Okay. I know I'm kind of beating this to death, but cardiac muscle. Cardiac muscle, see if I can blow this up a little bit. Okay, perfect. I'll get rid of my thing real quick here. All right. Cardiac muscles. There's still striations here. I got to blow a little hard for the striations. But everybody see these little lightning bolt lines, kind of right here, right here, right here, right here, right here, right here. All those lines, when you see that and it's striated, you know it's cardiac muscle. The difference between skeletal muscle and cardiac muscle is the presence of intercalated discs. The intercalated discs allow the electrical transfer of the muscle signal to allow all the muscles to contract simultaneously. The heart has to contract simultaneously, all the muscles. There's no ones that can hang out. When you pick up a pencil, your muscles are all contracting the same. When you lift 200 pounds, or when you lift a pencil, you're using different amounts of muscle contraction. Okay, the heart can't do that. You can control how many muscles contract depending on how hard you squeeze your fist together. Okay, you can't do that on the heart. The heart is all or none. Every muscle has to contract, and it has to contract at the same time. If they don't contract at the same time, you get an arrhythmia. Okay, you got to reset your heart. If the muscles aren't contracting at the right time, it's what's it called? Inversion. Okay, they'll actually go in there and stop your heart and then let it go again, reset it. That resets all the muscles contract at the same time. It's kind of inversion treatment. Okay? And it's to prevent the contractions from not occurring simultaneously. Okay, again, cardiac muscles. Let's see if this goes to it. Here we go. This would be more like something I show you. You can see striations, right? You can see striations. You can see intercalated discs right here, right here, right here, right here, right there, right here. Here's a big one. Okay, right here is that big one here. Okay, you can see the intercalated disc. Also, look at the nuclei. Remember in skeletal muscle, the nuclei was lined up along one line, right? Multiple nuclei in one cell. Here, the nuclei are scattered throughout it. So when you look at this, you'll see striated muscle. If you see striations, you know it has to be cardiac or skeletal. Okay? You see intercalated discs, you know it's cardiac. If you see all the nuclei lined up in a nice even row, it's skeletal. See all the nuclei scattered around? It's cardiac. Does that all make sense? Those are the differences. And I guarantee you'll see that on your final, on your midterm. I think on your midterm too. I'm gonna give you examples of a slide and I'll give you a nice picture. I won't give you something that you go, well, I mean, that's gonna be obvious. Okay? If you know what you're looking for. Okay, so those are the differences. Those I want you to see. Now, smooth muscle. No, we're not in smooth muscle yet. Okay, cardiac muscle. Everybody sees the striations. Okay, striations going up and down all through here. Everybody sees a nuclei. Here's one here, here's one here, here's one here, here's one here, here's a whole bunch, here's one here. They're not lined up. There's some somewhat lined up, but not lined up like skeletal muscle. Skeletal is all one nice little line. And look at this here. If you see a line like this, a line like this, like this, like this, you see these breaking lines. You can't miss them. That's cardiac muscle. Those are intercalated discs. That's what allows the muscle to contract 100% everything one time. They all contract simultaneously and allow blood to pump through the heart efficiently. Smooth muscle. Okay, now we're in smooth muscle. Smooth muscle is the exact opposite. Smooth muscle has no striations. Okay, that's it. This is your layer of smooth muscle. Look at all the nuclei lined up here. Okay, this is circular smooth muscle, so it means it's going in a band. This is longitudinal, means it's running this direction. So the longitudinal is running this way you're looking at. Circular is running this way you're looking at. They're running 90 degrees to each other. Okay, but you can see there is no striations in smooth muscle. Now, is smooth muscle voluntary or involuntary?
Involuntary.
Involuntary. That's what's around your heart. That's what's around your blood vessels, around all your organs, digestive tract, etc. Was cardiac muscle voluntary or involuntary?
Involuntary.
Involuntary. Perfect. The only voluntary muscle we have is skeletal muscle. Let's make this bigger. There we go. That's a good picture of it. Look here, this right there. Okay. Do you see any striations? You see lines going the longitudinal way, but nothing going crisscross. No striations. That's a circular layer of smooth muscle. That's what you want to look for. No striations. That's even a better picture of it. Because if this was skeletal muscles, you would see all these lines right here go in this direction. All these lines go in this direction. You don't see any lines. You see long, smooth, long muscle fibers here. All these long fibers with no striations. That's smooth muscle. You see them, and nuclei are splattered all through it. There's no organization with smooth muscle. Smooth muscle, they have nuclei everywhere. Okay? And no striations. Okay. Again, there's muscle tissue. No striations are apparent, and nuclei fly all over the place. Okay, the nuclei are not contained in any order, and there is no striations. Can I make that one bigger? No. Yes. No. Can't. Bring this back. Okay. And again, smooth muscle of the duodenum causes peristaltic action. Again, do you see any striations in that? You see a lot of fibers, right? That kind of muscle tissue has to be smooth muscle. There's no striations. It contracts around it like a concentric, like a circular motion. Okay, you can see the nuclei all spread out, and again, no striations. All right, perfect. So we did muscle, we did nervous tissue a little bit too. This we didn't do a whole lot of. We did that. So let's do at least nervous tissue. All right. Remember we talked about nerve. We talked about the soma. We talked about the nerves have different names. The cell body is the soma. The cytoplasm is called neuroplasm. And you have axons and you have dendrites. Okay. So this is a neuron scan here. Okay. And the axons are the one big one coming off of it. So here's your axon right here. Okay. Here's your big extension. That's an axon. That's an axon. This one here is an axon right here. Okay. But these are all neurons. These are nerve cells. And see, all these little ones here are dendrites. This is where all the information comes in. And the axon here sends the information out. So input information comes to all these little spikes here, all these dendrites, goes into the cell body and gets voted on. If it's a stimulation high enough, if it reaches the threshold, then the axon fires a signal and it goes through the axon, which is a large one here. And the nerve cells are the largest cells in the human body. Some of these axons can extend up to 3 feet in length. So they can be long. Okay? So you have axons, you have dendrites, and you have the cell body, which is your soma. Okay? Again, you can see your neurons. Again, these are your dendrites, the smaller ones here, and this long one here is your axon, where the information travels. Input comes into the dendrites, input goes out of the axon. Okay, here's a bit bigger one, which is good. Okay, again, these are your dendrites that line up. See all these little, get out of there here. I can't do that. But all this information, these are all feeders. Information comes into all these dendrites. And this big thick one here comes off. This is your axon coming off the cell body. Okay, again, here's your axon. You can see this big, thick portion here. So here's your axon coming off. And these are dendrites that receive information. These here, these here, these here, this here. Okay, so dendrites are a bunch of little ones. Information comes into those. The cell votes on it. If it wants to send it on, it goes through the axon and goes to the other cells. Okay, you also have what's called neuroglia cells. These are cells that support the nerve cells. They're not neurons. They do not transmit information, but they support, allow the nerves to carry on their function, bring them nutrients. Because remember, you can't replenish nerve cells. Once a nerve cell dies or it's damaged, that's it. Okay, you're born with as many nerve cells as you're going to have. You cannot make more nerve cells. They cannot regenerate. Okay, this is a big blown up picture. I'm going to have to take a while to load this one. All right, so these are all the dendrites. This is where information comes in. What are all these little blue dots around it? Those are your neuroglial cells, like right here, right here, right here. These are supporting cells. Some of them are phagocytic cells. Some of them make the blood-brain barrier, which we're going to talk about. Okay, those are neuroglial cells. They isolate the neurons and protect the neuron cells. They make sheaths around it so it can conduct impulses quickly, called myelin sheaths. Those are all the support of the neuroglial cells. The neurons actually transmit the information. The neuroglial cells support the neurons. Okay, this is your cell body. There's a nucleus here. You can see the nucleus in the cell body. And this part, this is where the axon leaves the cell body. This gets real thick right there. If you take a look at it, without this blue thing right here, it gets real thick. That's called the helix, which is where the neuron assembles everything and then makes the axon. Okay, so this is a neural helix, or the axon helix, and then this is the axon. Okay, you look at this one here. These are nerve cells. Again, you look at it, this is the cell body. And then here's the helix right here, where all the nerve cells come around, and this is your axon as it travels. And it travels through the cell. And all these other ones back here, these are dendritic extension, or dendrites, where information comes in. And all these little dots, these are neuroglial cells. They form barriers, they form sheaths, they protect the neuron, they eat pathogens that come around the neurons. They make sure the neurons stay healthy and fire. But they do not have a nerve impulse themselves. So neuroglial cells do not carry an electrical impulse. Only neurons do that. Okay. Okay, so this is nerve fibers. You can see how they're all composed. All these dark areas here. Okay, this is a myelin sheath. The myelin sheath is made by some of the neuroglial cells, and it protects the nerve and it makes the nerves travel extremely fast. If the nerves do not have a myelin sheath, it's like a tenth of a second. If the nerve has a myelin sheath, it's in milliseconds. I mean, 1,000 times faster. Okay, there's areas in the nerve that where the sheath is split, it doesn't combine. If you look here, I know it covered over it just right there. What else? There we go. This is myelin. This is made by the neuroglial cells. Myelin is the nerve sheath that surrounds the nerve, allows it to transmit extremely fast. Homer Nichols jealous, aren't you? Okay. Now some of those, look right here, to the right of the arrow, you can see where there's a little gap. That gap is called nodes of Ranvier. That's where the myelin sheath is not continuous. So the nerve impulse has to jump across that. Okay, those are nodes of Ranvier. Okay, and you'll see them every once. You'll see one right here. You see ones without the blue stuff in the way. Okay, that's the best one right there. See it right there. Okay, those are where the myelin sheath is not continuous. Okay, but all your fast nerves have a myelin sheath around them to conduct that electrical impulse extremely fast. Okay, nerves are just like muscles; they're in fascicles. Okay, that's a cell. The fascicle is a group of nerves. Okay, that's how they run. Okay, the perineurium, just like the periosteum is outside the bone, the perineurium is the outside of the nerve cell. Okay. And again, this is a fascicle. It's a little bit bigger view of it. Okay. This is a perineurium. This is the outside of it. And this is your nerve cell. This is your fascicle. This is a whole collection of nerves that run through each bundle, like a nerve bundle. Again, we'll go into a lot greater detail when we go into all the different nerves of the body. That's all they want to know. We're afraid to ask. Okay. Again, you can see the myelinated nerves. All the myelinators have, let me get that off of there real quick too. Okay. And then you have the nodes of Ranvier, which are the separations where the myelin breaks. You have the myelin nerve fiber that surround it. And what makes myelin is what's called Schwann cells. Now, the Schwann cells play a really important role. We used to think nerves could not be repaired at all. Nerves cannot regenerate ever. If you lose a nerve, you've lost it for life. If you damage a nerve, you can actually get some repair now. This is called Schwann cells. Schwann cells initiate a type of bridge that allows the nerves to actually communicate one side to the other. A lot of times when we do wisdom teeth, we sever the nerve that runs through the lower part of the jaw because of the teeth wrap around it, and we damage the jaw. Okay? And the person becomes numb on their lip for the rest of their life. There are some cases where the Schwann cells can regenerate. They don't regenerate a nerve, but they allow a connection to form between the two severed ends, which you never thought was possible when I was taking anatomy, which was a long time ago. Okay, so now it doesn't make a new neuron. Don't get me wrong. Okay, but it allows the two ends to actually connect and form a pathway that allows them to talk to each other again. And that's real important with Schwann cells. Okay, they allow that to happen. They produce that myelin layer. And again, we'll go over that in real great detail when we do the section on nerves.
There's a question in the chat.
Oh, Schwann cell similar to a stem cell? No. Because a stem cell, that's a really good question. That's a great question, actually. A stem cell divides and multiplies and makes a new cell to replace other cells. A Schwann cell allows two ends of a severed cell to communicate with one another. Does not make a new cell. Nerve cells can never regenerate. They can't use stem cells. Okay, so stem cells actually form a new cell. Schwann cells actually form a connection or this myelin sheath and helps the nerves, the severed ends, to be able to communicate. Does that make sense? So don't get Schwann cells confused with stem cells. It almost sounds like they are, but don't. It's a different function. Does that make sense? Cool. All right. Okay. Oh, no problem. So you got the epineurium, which is around it. You got the perineurium around each fascicle, and you got the fascicle here. So here's your fascicle. This is a nerve. This is one nerve in cross section. Around it is your perineurium. That's the outer layer. Okay. Around that is the epineurium. So the epineurium, the perineurium, and then your fascicle. That's how the nerve bundles are designed. And yeah, I know we haven't talked about that yet, but we will get into that. Okay. You can see your myelin sheaths. These are the cells that make the myelin sheath. Again, the endoneurium is on the inside. These are your axons. You can see it's cross sections are the most difficult things to read in human anatomy. Most physicians cannot read cross sections. You can see an MRI, a CAT scan, those are cross sections. And most people can see obvious things. It has to go to radiologists. They read it and they tell the physician what they're looking at. Okay, then they can tell you what it is. But if you look at this little dot right here, okay, that's an axon. These are axons. These little dots right here. Those are all axons. Do those look like axons? They're little dots. That's the axons running this direction, though. You're seeing the end of it. I do not expect you to be able to identify cross sections, except for real basic stuff. I would not expect you to know that as an axon. Those little dots right there. Okay. This is your endometrium around it. Those are your axons. This is the perimetrium around that. And this is your fascicle of your nerve cell. And this is the myelin sheath. You can see these little areas right here. You can see the axon inside. The blue area is the myelin sheath. The little dot inside is the axon. That what surrounds it. Like I said, don't panic if you can't see these cross sections real well. They're extremely difficult. Any part of the body. Cross sections are extremely because you don't see the long section. You don't see the organ. You see a cross section of it. And it takes a long time to read those correctly. Now, this you can see. You can see a Schwann cell right there. You can see a Schwann cell right there, right there, right there. You can see the Schwann cells in a longitudinal section. Okay? You can see the nodes of Ranvier. Look at, you can see, if I can get that off of there. Let's go there. Right there. Let's get around it. Okay? So if we go right here, you can see this constriction of the nerve right here. You can see how it comes together. That's a node of Ranvier. That's where the myelin stops. There's a gap between the myelinated section. Those are called nodes of Ranvier. Okay? And that's where the nerve fiber jumps across that space. And these are all myelinated fibers. You can see all the myelin here. Okay? This is in the cerebellum of the brain. Cerebellum has all these grooves, and we're going to learn the cerebellum is your coordination center of your brain. That's the used to be called the second brain. It's the lower part of the brain just above the medulla oblongata. And it controls, my daughter used to call it your cool points. You know, when you walk and you trip over your feet, you lose cool points. The cerebellum controls you be able to walk and not stumble, not fall. Okay, that's your cerebellum. Okay. It has sulcus, which are grooves. When you look at a brain, it looks like cauliflower. Okay, it's got gyruses and it's got sulcine grooves. A sulcus is a deep groove. A groove is a narrow sulcus. Gyrus are bumps, the bumps in the brain. We'll look at that. You have white matter and you have gray matter that run through the brain. And you'll be able to see that when we do the dissection. We'll be able to see when we do dissections on the screen here. Okay? The gray matter is your cell bodies. That's where all the information is processed, voted upon. That's what kicks all the information. Your cell bodies is your thinking portion. The white matter is all your axons, everything that travels through there. Because myelin's white. So all your nerves are white matter. So white matter is your myelin. Gray matter is the cell bodies that make up the nerve. That's your thinking portion where everything gets evaluated. Okay? And you can see on here, you see the sulcus, which is a really deep groove. And you see the gyrus, which is a bump. It's like a light show here, huh? You can see the gray matter because it's gray. This is your gray matter. It's where all the cells are. And this is your white matter. This is all your axons that are traveling there. And again, we'll go that way over the brain. We got a lot of sections on the brain. Again, different stain on it. This is your sulcus. Sulcus just means a groove. Gray matter is your cell bodies, thinking portion. White matter is the axon that transmits the information. Okay, main part of the brain is right there. Now you know everything there is to know about the brain. Not by a long shot. The brain's got the most complicated sections. Okay, sagittal section cut through. You have something called Purkinje cells in the cerebellum. They're unique to the cerebellum. Function's not real clear, but they're very distinct in what they look like. You can't really see them right there because everything's covered with this stuff. There you go. But these are your Purkinje fibers right here, Purkinje cells. And this is your gray matter, all your cell bodies. Spinal cord. Spinal cord has a lot of white matter because that's where all the cells run. You have different horns of the cord we're going to look at. We're going to look at the ventral, which is your front part. We're going to look at the dorsal, which is the back part. Okay. You have a dorsal median sulcus, which means, think about it. Dorsal is back. Median means the midline. Sulcus means a groove. So there's a groove on the back of the vertebral column in the middle. Dorsal median sulcus. Okay, this is a dorsal funiculus. This is a group of nuclei that vote on things, that make you think in different areas. You have a gray commissure, okay, which is your cell bodies. You have a central canal. The central canal is where your axons run through the canal there. That's where all the information is transmitted. You have a ventral median fissure. Again, ventral is front surface, median is midline, fissure is a groove. Okay, fissure and sulcus are both grooves. And you have a ventral funiculus, which is a voting section, which are cell bodies. You have a ventral horn and you have a dorsal horn. Ventral horns on the front surface, dorsal horns on the backside. And you have a lateral funiculus. So lateral funiculus on the side, ventral funiculus on the front, dorsal funiculus on the back. All right, again, dorsal funiculus is a group of cell bodies. Dorsal horn is the back. Ventral horn is the front. Lateral funiculus is the side. Lateral means to the side. Medial means to the middle. Central canal, where your axons run. The gray commissures, the gray means cell bodies. And then your ventral funiculus. Ventral means the front. Dorsal means back. All right, spinal cord again. Big blown up section here. Gray commissure, you can see all the cell bodies in there. Central canal, that space where your nerves run, where your axons run. Ventral horn, dorsal horn. Okay, I want to say this wrong. Dorsal horn is your input. Ventral horn is your output. The afferent and efferent nerves coming in. We'll go through all that too. These are your cell bodies. You can see all the nuclei, and you can see all the different cell bodies inside of here. Okay, as you go down through the spinal cord again, down by the thoracic version of it, we start out the same things. You got the lateral horn, you got the dorsal horns, you got the dorsal funiculus, the ventral funiculus, central canal, and the lateral funiculus. Gray commissures, okay, is where your cell bodies are. Your axons are running through the central canal. All right, spinal cord again down the lumbar region. As it's getting farther away from the brain. The dorsal funiculi, same basic pattern. The dorsal medium sulcus, central canal, ventral horn, dorsal horn, ventral medial fissure, ventral funiculus, and the gray commissure, and the lateral funiculus. Same structures as you're going down the spinal cord. All right. This is a different stain, so it looks a little differently, but you can see it. You can easily see the central canal. Okay, this is a cross section. I would expect you to be able to identify the central canal as cross section. It's in the center and it's hollow. The gray commissure surrounds the central canal. That's where your axons run. This is where your cell bodies are. This is your ventral median fissure. This is your dorsal median sulcus. They both grooves. This is a ventral horn. This is a dorsal horn. This is towards the front of your body. This is towards the back of your body. Those structures are easy to identify. Again, very easy to see the central canal. Okay, there's your central canal. Your gray commissure surrounds the central canal. The gray commissure is your cell bodies that receive those input from the axons, from the central canal. Neuroglial cells. What do we say neuroglial cells were? Do they conduct electrical impulse? No. It's perfect. They're the supporting cells. They surround the neurons and they support the neurons so the neurons can conduct electrical impulse. The only cell that can conduct an electrical impulse in your body and transmit information is a neuron. Okay, these are somatic motor neurons. These are your cell bodies. Okay, these are dendrites here that surround it, and the axon would be the thickest one. It's hard to tell which one's the axon here, but this is probably the axon because it has a helix here and extends down. But I'll make it easy when we tell because dendrites are usually smaller and thinner, and axons are thicker. Okay. And dorsal root ganglia. Here we have a dorsal section and a ventral section. This is a dorsal section. This is where output comes through. These are called satellite cells that surround the neurons. And these are the cell bodies of the unipolar neurons. Unipolar neurons direct information in only one direction. You have neurons that go both directions. These are different types of neurons. We'll go over all those, but these are unipolar neurons. Okay, you can see this, the histology of those. And again, satellite cells, you see surrounding the neurons. Okay? And these are your neurons right here. And these are your satellite cells that surround it. And the satellite cells make that myelin sheath around here.